Begin in the Rosalind homepage. Navigate to your project folder containing your experiment of interest. Then click into your experiment. After your experiment has been launched and processed, the full results are organized and presented in 5 tabs at the top of the page. The Experiment Summary tab displays the experiment title, description, and setup parameters. Additionally, a description of the methods used to analyze your data is presented at the bottom of the page. These methods include relevant citations and can be adapted for use in publications. The Quality Control tab, represented by the table icon, contains a table with well-established QC metrics that assess the performance of sequencing for each sample. You can hover over the title of each column to display additional information about each metric. Any metric value that falls outside of the preferred thresholds will be flagged with a note on the far right side of the row describing the issue. As you can see here, all samples have passed QC. On the left side of the page, there are several figures that provide additional insight into the quality and performance of your samples and experiment. The QC figures for bulk RNA sequencing include a sample correlation heat map, a violin plot, a genomic region plot, an MDS plot, and a gene body bias plot. Clicking on each figure will open a window with details about the visualization and options for download. The sample correlation heat map provides a visual representation of the Pearson correlation between samples and clustering based on those correlations. Figures are annotated with attribute information to facilitate assessment of similarity within biological replicate groups or to help identify potential outliers. Click Resize to view the image in a larger frame, or click Download and select your preferred download format for the image. A CSV file containing the correlation values can also be downloaded. The violin plot displays the log distribution of the unnormalized gene counts for each sample. Click on the additional image on the bottom left to learn more about interpreting this plot. The genomic region plot shows how mapped reads were distributed over gene features across the genome. The MDS plot is a visual representation of sample similarity mapped on a 2-dimensional space. Click on the additional images on the bottom left to visualize this plot according to your different attributes. The gene body bias graph shows the distribution of mapped reads over gene bodies, averaged over all expressed genes. Different library preparation kits will yield different characteristic shapes. For example, some kits may be biased towards the 5' end, while others may be biased towards the 3' end. Look for a similar pattern between the samples. The Samples tab, represented by the test tube icon, will bring you to a page listing all samples in the experiment and their corresponding attributes. It is essentially a summary of the experimental design. You can view the information in a standard table, or you can use the Grouped and Colored by drop-down menu to organize and color rows of replicate samples within the table. Here, if you're the experiment owner, you can add additional attributes that can be used to set up comparisons for differential expression analysis. In the File Management tab, represented by the cloud icon, you will find the files associated with your experiment, including attribute files, FASTQ files, processed counts files, and differential expression results files. Thanks for watching.